Hi, my name is Lindsay Potts, and this is my presentation on the paper titled Massive Expansion of Human Gut Bacteriophage Diversity, which was published in the journal Cell in 2021. And I hope you enjoy. So first of all, I just want to get into the bottom line up front here, which is demonstrated beautifully by this graphical abstract that was provided by the authors. And basically what they did here was they took 28,000 human gut metagenomes from 28 different countries, and after sorting through this data and cleaning it up, they were able to assemble contigs for 142,000 non-redundant phage genomes. And then they sorted through these phage genomes, and I want to clarify that viruses are classified based off of their genome type, but here this isn't really an issue because bacteriophages all have the same genome classification, and then they're classified based on their host species, so that's what they did. And then because they had metagenomes from these 28 different countries, they were able to do some epidemiology and kind of figure out what phage species that are present in the gut phagome were associated with which geographical locations. And then they actually discovered a new clade, which I also want to clarify because I don't think I put it on the slide. When I refer to the word clade, it just means a taxonomic group. But they discovered this new clade, this new taxonomic group of bacteriophages present in the human gut, which they named the gubophage clade. I just want to cover a little bit of background before we dive into the bulk of the presentation here, and I want to preface all of this by saying that phages are by far the most diverse as well as the most numerous group of viruses that exists. And I also want to point out that when I say phage in this presentation, I am specifically referring to bacteriophages, even though there are also phages that infect archaea, as demonstrated by the phages in green here. So a little bit more background. Like I said before, bacteriophages are the most abundant as well as the most diverse group of viruses. Viral metagenomics, since its foundation in around 2002, has led to the discovery of hundreds of thousands of new phage species. And crossphages and cross-like phages, when I say that, I am referring to the most abundant group of phages that exists in the human microbiome, and they were discovered in 2014, and the name CRAS comes from the cross assignment software that they were discovered on, so that's why it's capitalized like that. And also I want to say that when I say the word phagome, I am referring to the sum of all phages in an environment. Just like a virome is the sum of all viruses in an environment, and the microbiome, as you should know at this point, is the sum of all microbes in an environment. So what is the need here? Why do we care? And why do we want to know about the diversity of phage species present in the human microbiome? First of all, the human virome has been shown to outnumber the human microbiome about 10 to 1. So for every microbe that is present in the human body, there's about 10 viruses. And that alone, I feel, is enough to warrant study over this topic because we want to know what's in our bodies. On top of that, the majority of gut phage diversity remains uncharacterized and incomplete. There was a significant review published in Nature in 2020, which goes over this, as well as pretty much the field of phage genomics up until this point, so around 18 years of phage metagenomics. And for what? On top of that, the gut phagome has a major role in shaping the gut microbiome because bacteriophages are viruses that infect bacteria, and bacteria also pick up a lot of genes from bacteriophages. A great example of this is the CTX phage, which infects Vibrio cholerae. And without the CTX phage genes, Vibrio cholerae does not actually produce an enterotoxin and cause cholera. So this enterotoxin gene is picked up from the phage. But even though the gut phagome has a major role in shaping the microbiome, because we really haven't looked into it further, its link to illness is unknown. And if it does have a significant link to illnesses that are associated with the gut microbiome, then that could possibly provide insights into prevention and treatment. So the research question that we're trying to answer here is super simple, super upfront. What bacteriophage species are present in the human gut based on the metagenomic analysis that was conducted here? For the methods involved in this massive metagenomics study, they obtained 28,060 sequencing reads of human gut metagenomes from the European Nucleotide Archive. And these sequencing reads were obtained via Illumina sequencing, as this is the gold standard for metagenomics. Paired end reads and single end reads were assembled using two different softwares, those softwares being Spades and Megahit. And then they predicted viral sequences using Vir Finder and then Vir Sorter, which I am very excited to try out because I just found out these existed via this paper. After that, they just used simple blast searches to decontaminate these sequencing reads from any human DNA that could be present because, as most of you should know at this point, when we're looking at human gut metagenome samples, it's usually from human stool samples, so we need to decontaminate any human DNA that may be present. After this, they were able to cluster sequences using CD-HIT and do quality control over these predictions using a bunch of different softwares that I will not get into because of time, but essentially did a lot of quality control to remove low quality reads, short reads that would be too short to constitute the identification of a phage genome, as well as the removal of genomes that were predicted to belong to non-phage taxa so that we are only left with phages. After that, they were able to start the taxonomic sorting by clustering phages into VCs, viral constructs, so essentially assigning a viral construct to each of these sequencing reads. We're able to do taxonomic assignment then using HMER, which is again just kind of a simple standard taxonomic software, as well as the ViFog database. They did host assignment using CRISPR spacer sequences and CRISPR-Cas5 2.0 from the 2,898 gut bacteria that are used in this CRISPR-Cas5 2.0 software. And they assigned host predictions based off of the output from the software. They then did taxonomic assignment of bacterial genomes using the Genome Taxonomic Database Toolkit, and proteome analysis and functional annotation of these different reads that we had here, these different VCs at this point, with a software called Dustmasker, and then they, as well as putting them into BLAST of all the protein sequences and running the Markov clustering algorithm, and removed clusters with only one member, essentially with one protein, because you can't really have a virus with one protein. There has to be some kind of proteome there. After that, they did metagenomic read mapping to estimate the prevalence of each viral species and looked into the geographical distribution of metagenomic samples by removing samples with a sequencing depth of below 50 million reads per sample. And after this, they were left with only 23 countries instead of the original 28, but they still had all the continents that were originally included. Similarity between two samples was calculated by computing the number of shared VCs divided by the number of VCs in both samples, which is called the Jacquard index. After then, which we will get into with the gubophage, the new clade that they were looking into here, that they discovered, the phylogenetic tree comparing gubophage against crast-like phages was constructed by aligning the corresponding large terminase genes with MAFT, which we will talk about a lot more in the results section and discussing the results, trust me, followed by FastTree in our studio. I've used FastTree before. They identified gubophage clades by calculating the fraction of shared protein clusters among the gubophage genomes and then carrying out hierarchical clustering with average linkage and Euclidean metric. Genome-wide comparisons between the gubophage clades were generated with EasyFig v2.2.5, which I believe is also in our package, using the T-BLAST algorithm. And then they just did some basic quantification and statistical analysis for the epidemiology portion to kind of really validate their geographical distribution analysis. Section was a lot, but buckle up because now we are on the results section. It's about to be even more. I would just like to remind you all quickly that we are on part A of the results. As you can see here on this second slide, back in the graphical abstract, the results in this paper were kind of divided into 3 separate parts. So part A, B, and C, that's just what I'll refer to them as. And yeah, we have the host assignment, worldwide epidemiology, and the discovery of this new gubophage clade. But anyways, let's get back to our results section. So as you can see here with the bacterial host assignment, host prediction results, by far the most prevalent phylum of bacterial host species here was Firmicutes, as well as Bacteroidota, Actinobacteria, Protobacteria, and a little bit of Campylobacter, which is nice because those are easy to pronounce. But then when we look at the genus level, especially of all these Firmicutes here, analysis at the bacterial genus level revealed that across all phyla, revealed that Lactinospira, Rosaburia, Aganthobacter, Prevotella, and Bloscia A contain the highest number of VCs per isolate, with the exception of Prevotella, which belongs to the gram-negative Prevotellinaceae family. These genera are members of the gram-positive Lactinospiraceae family of Firmicutes associated with butyrate-producing spore formers, which I don't know much about butyrate-producing spore formers, but that does not sound like something I would want in my gut microbiome. And interestingly enough, too, in contrast, the lowest viral diversity per isolate, so down here, was detected among Helicobacter and lactic acid bacteria Lactobacillus H, Lactobacillus, Enterococcus and Pediococcus, which from my knowledge of the other presentations I have watched in this course, seem to be a lot more prevalent and a lot more numerous, some of the most numerous bacterial genre in the gut. So this is actually also suggesting a commensal relationship of some sort where these phages might actually be helping us and killing off bacteria that we may not want in our gut microbiomes, which is super cool. And this is also just kind of a tree demonstrating this, a weird circular tree, but a tree nonetheless. Host assignment was carried out by linking prophages with their assemblies in the CRISPR database that I mentioned back in methods a minute ago. Orange connections represented VCs with a very broad host range, not restricted to a single genus, which is the other thing I wanted to talk about here. The host range of different bacteriophages has not been really investigated at a large scale, especially because hundreds of thousands of new phage species have been discovered, and it's just impossible to look into that many phage species, but they did here a little bit. And the bacterial taxonomic ranks here revealed that the majority, around 64% of VCs, were restricted to infect a single species, but they also found many VCs with a broader host range, such as those restricted to a single genus, which was 22%, family 10%, order 1%, class 0.26%, and phylum 0.13%. So very interesting. Findings happen to be in line with a recent survey of host range of gut phages that they cited in this paper. That around in the paper they cited, they said that 69% around of gut phages were restricted to a single species. So that is consistent. Visualization of the broad range VCs reveals that the large-scale connectivity between distinct bacterial species could be fuel for bacterial adaptation, evolution, again, co-evolution of phages with bacteria. That happens a lot because they are vectors for horizontal gene transfer. And in general, the higher the viral diversity per bacterial genus, the higher number of phages with a broad host range. So the more diverse the bacterial genuses may be, the more likely that bacterial genus was to have a phage that had a broad range. Surprisingly, 2 VCs had a host range that spanned 2 bacterial phyla, which is interesting. And these were predicted to infect Faecalibacterium prosynetizini, I'm definitely pronouncing that wrong, and a 2 Bifidobacterium species, whereas VC, the other one, had a host range that included 5 bacterioid species and Bloschulae wexlerae. VC269 was predicted to be a myoviridiophage, and they could not assign a taxonomy rank to the other VC that they had here. That is part A. Here they found that approximately 1/3 of gut phage had a broad host range, not limited to a single host species. The rest of the other 2/3 had really a single host species here, that 63, I believe it was, percent. Oh, now on to part B, the human lifestyle associated with global gut distribution of phagome types. That's what this section is titled. And they essentially, so now on to part B of the results, the geographical distribution part. They ended up observing clear separation of the North American, European, and Asian phagomes from African and South American samples. And I want to point out that even though later when we're talking about the gubophages and cross-leg phages, we're talking about westernization, here there's kind of a clear divide when it comes to the industrialization and economic development of these countries, which supports the hypothesis that the human microbiome is largely associated with lifestyle factors such as urbanization. And they said that country-wise, samples derived from Africa and South America were mainly sampled from Peru, Tanzania, and Madagascar. Specifically, Peruvian and Tanzanian samples originated from hunter-gatherer communities, whereas Malagasy samples came from rural communities with non-Western lifestyles. And Oceania was also a special case here because it had a similar fraction of samples belonging to both groups, rural and more urbanized. However, when they separated it by country, they ended up revealing that all Fijian samples clustered with the rural group, whereas Australian samples segregated with the urbanized cluster, which is kind of what we see in the trend of how these countries operate. So here we have a principal component analysis plot. So I just want to provide a little bit of a segue into section C of the results before we get into it, just to kind of explain the author's methodology of thinking and how we got to the discovery of this new bacteriophage clade. And I want to mention also that there was a figure associated with this, but I did not include it because it doesn't really have to do with this specific study. It's kind of more about the literature, and also it's just very confusing, but I still want to talk about it. So as I just mentioned, they observed here and kind of hypothesized that the human gut phagome could be influenced by the population of the microbiome, and it is also largely hypothesized that the microbiome, one of the big factors that influences the composition of the microbiome, is lifestyle. As we see here, we have more urban lifestyles and then more agrarian rural lifestyles. And they looked into the literature and discovered that the Crass-like family of gut phages, which can be divided into 10 genera, also kind of had this clear divide, where genus 1 was found in a large fraction of Western microbiome samples, whereas the other genre were found in more rural agrarian populations like this. So then they further investigated this, if they could take the VCs from this metagenomic study that they had already done and kind of apply that same thinking. So they took these 28,000 VCs and were able to detect a total of only 280 VCs that were globally distributed, so found on all 5 continents, or at least 5, they said, 5 out of 6, which 280 out of 28,000, that only represented 1.3% of all defined VCs. For 119 out of these 280 VCs, out of this 1.3%, they were able to classify them to be the Caudoviralis order, and the remaining 57.5% were unclassified, which is pretty significant, obviously. When they looked at the viral families within Caudoviralis, they saw Podoviridae, Myoviridae, Cyphosviridae, and the newly formed family Horellaviridae. And I also want to mention that the Crass-like phages, as well as the gubophages, which I'm about to talk about, are all part of Podoviridae. A bacteriophage network of globally distributed VCs revealed that Prevotella was the most targeted genus, followed by Faecalibacterium and Rosbura with 15 VCs each, which have also been kind of shown to be globally distributed in the gut microbiome as well. So that kind of goes with this hypothesis that the gut microbiome could affect the virome, phagome. And when they were looking to see how many of these were part of the Crass-like family, there was a little bit of disconnect there. So they decided to dive into this disconnect because they found essentially that although 12 globally distributed VCs were members of Cross-like family, they were only able to assign a host to 6 that targeted the Bacteroidetes, which is pretty much what Cross-like phages target. So that's where they were kind of like, hey, there's probably another taxonomic group here. Annotation of the goobophage genome revealed that the goobophage is organized into 3 distinct regions. One region encoded structural proteins, the second was comprised mostly of genes involved in DNA processing, and a third encoded for a series of hypothetical proteins, which was just the construct. And once again, analysis of the distribution of the goobophage clade, as we see here, revealed its presence in all the continents except South America. And particularly, it reached a prevalence of close to 40% in Europe, and the lowest corresponded to Africa. The discovery of this clade is another example of a highly prevalent phage in the human gut, and highlights the need to perform further culturing and mechanistic studies to better understand its role in the human gut microbiome. So, just to summarize all of these results that we just went over, they discovered this new clade of bacteriophages and named it goobophage. And on top of that, goobophages and cross-like phages they found are mostly present in the Western industrialized gut. What are some future directions that this study points to here? Number 1, wet lab experiments to determine phage pathogenicity to see if they are actually affecting the bacterial species present in the gut and the bacterial hosts that are proposed here, as well as the humans that these phages live in, as well as coevolution, because like I said, bacteriophages are huge proponents of horizontal gene transfer and often engage in coevolution with bacteria, as well as further characterization of elements of phage genomes to really figure out what's going on here. I also want to point out, even though it's not on the slide, that this is also huge, especially the pathogenicity part, because when we are looking into phage therapies, which are kind of one of our biggest hopes for overcoming antimicrobial resistance and having an alternative to antibiotics, we do really want to consider phage pathogenicity, because we don't want to be affecting the gut microbiome, or we want to leave it alone as much as possible, and we really would not want to contribute further to human disease in any way when we are coming up with a possible novel antimicrobial solution. So, once again, conclusions here. Goobophages make up a significant portion of the bacteriophages species present in the human gut. Goobophages and cross-like phages are largely associated with Western and industrialized microbiomes. And here I just have some questions that I've put up for discussion. First of all, why are goobophages and cross-like phages associated with Western societies? Is it dietary or is it more environmental? Could the virome play even more of a role in disease than the microbiome? And what considerations should we take regarding the human phagome when it comes to development of phage therapies and antibiotic alternatives? And here is my reference slide. Here is the paper, and here was the Nature review that I was talking about.